And then I'll go on to introduce our speaker. Sorry, there's a fly like right in front of my face right now. We are so excited to welcome back Jason McLellan. Professor McLellan is a professor in the molecular bioscience department at UT, where he researches viral and bacterial proteins. His work to stabilize the spike protein is found in all the major COVID vaccines in the US, as many of you are probably aware. His research continues to be on the cutting edge of protein engineering for vaccines and antibody therapies. And recently his work around the stem of the spike protein, a part of the spike that is highly conserved between coronaviruses, has been highlighted. I recently read an article about it in Science, as it could pave the way for pan-coronavirus vaccines that would be durable against variants, as well as different emerging coronaviruses. We're really excited to be able to speak with him today and ask him some questions that have been on top of our minds related to vaccines, antibody therapies, variants, global vaccination efforts, and boosters. And so I'll turn it over to Jason to give some initial thoughts, and then I will facilitate a Q&A. And one thing I'm going to do before I put my articles in the chat is I'm going to put a link to Slido, which will allow you guys to submit your question and answers and then vote on others. So.
Cool. Caitlin, thanks so much for that very warm introduction. Happy to be back with all of you. And yeah, just thought we could chat about whatever is on your mind and whatever information I have to help you out. Obviously, I think we're all kind of concerned about the increases in Delta. And I'm sort of curious about your modeling efforts too, and how you model maybe two discrete populations of human behavior. But anyway, yeah, happy to open up to the forum and let's chat.
Does anyone have, yeah, I was saying we'll start with start with just going shooting.
Okay, so yeah, would you give us your take on the Johnson & Johnson headlines from this week? I mean, should everyone with the Johnson & Johnson go out and get whatever second shot they can get their hands on as quickly as possible? And
absolutely. Yeah, I would say
boosters in general, where are we going with boosters?
Yeah, okay, yeah. So for J&J, I would say absolutely get a second shot if possible. I mean, even back then, it was kind of hard to say, you know, all the vaccines are really great. All three of them are the same, because J&J really isn't as good as the mRNA vaccines. I think that's clear. And or maybe it is if you gave 2 doses. I mean, it's essentially a half-finished product in my opinion. I think the efficacy from 1 dose of J&J would probably be similar to the efficacy of 1 dose of the mRNA vaccines, but for marketing and other reasons, J&J is just pitched as a single dose vaccination. And so I would strongly encourage anybody to follow that up with a boost of the mRNA vaccines. I guess we don't really know about boosting again with J&J. I mean, there's some concern about vector immunity and the fact that maybe you already then have antibodies against the Ad26 vector. Believe they have a phase 3 clinical trial underway examining a prime and a boost, but I don't know the results of that. I think like the New York Yankees have all been vaccinated with J&J, and they've had over like 12 players get infected again. So I think real world shows it's not that great.
Yeah, anecdotally, I'm in that trial, so I got 2 J&J.
Oh, so. Okay, awesome. How does the second one feel?
So I had no symptoms, nothing at all to the second one. Yeah.
Okay, cool. Yeah, 2 separate outbreaks for the Yankees. The first one was a couple months ago, and then a more recent one now.
And where are we headed with boosters for the rest of us who already have our double mRNAs?
I don't know. Clearly, Pfizer wants to give boosters for sales. It is hard ethically to justify giving a 3rd dose to us when 90% of the world hasn't received a first dose. So there's like an ethical consideration, a scientific consideration, of course. I mean, if vaccine was unlimited, should we get boosters? I know I'd feel better with a boost. I think we're starting to see data, some of the data looking at 250 days after the second shot, antibody titers starting to wane a bit. And we never intended the vaccines to cause sterilizing immunity, right? Like, we are trying to protect against serious disease, hospitalizations, and deaths. And so I think, without the third booster, we are seeing people get infected again with Delta, but really not having too severe. The problem is that is almost a perfect breeding ground to select for Delta to evolve further, for getting active replication in people with good antibody titers. That's probably kind of an ideal situation to allow it to escape further and make a 0.3 or a 0.4 variant that has many more of the escape mutations in it.
So do you think, based on what you've seen, the efficacy of the vaccine, like when you think of boosters, are you thinking of the same shot we all just received, or are you thinking of a new kind of one that's targeting a specific variant, and which will happen quicker, like the virus evading the immunity or immunity waning?
Yeah, that's a great question. So I guess in terms of boosters, I was thinking more of just a third dose. There was data from Moderna comparing a third dose of mRNA 1273 versus a third injection, but a first dose of a spike protein that can you hear me? First, a spike protein from the South African, the B1351. And the third dose of mRNA 1273 actually worked really well. Not as good as the third injection, first dose of the B1351 spike, but that saves a lot of manufacturing. And then you have these like two different things on the market, and how many times are we willing to reformulate? So I think just a third dose of the same one would be good for now. And the B1351 has many more, I think, escape mutations than does the Delta variant. The Delta doesn't have as many, so I think that'd be reasonable to boost with the original.
And if we boost with a different formula for a different variant, does it have to go through a different trial again, or is it just assumed that it's pretty much the same as the original one?
Yeah, I've talked with BioNTech about this because they're thinking about using one of our second gen spikes. And they don't think it has to go through, like they can make a handful of changes. I don't know what the number, but they can make a handful of coding changes and not have to go through all the clinical trials again. In the same way that we don't make the yearly flu vaccine go through a different set of clinical trials.
Yeah, my question is kind of around that question. Do you see a world where we're kind of updating these vaccines every year, every 2 years, or even every less than a year, with a combination of different spike mutations and escape variants?
Yeah, I mean, that was the question I was asked back last year, and I always felt like we were going to have to update at some unknown frequency, which I think is still unknown. Whether it's yearly, biannually, every 5 years, I think we're still waiting to see. And it probably really just depends on what the virus does, and if it evolves a variant that really escapes from these initial set of vaccines. B1351 was pretty close. I mean, that was showing like an 8 to 10-fold decrease in efficacy, but fortunately that one didn't really take over. In a lot of ways, we were kind of lucky that Alpha took over, because Alpha doesn't escape the vaccines really well. So I think it'll just be something that follows, but it does lead to then, like, you'll have so many different people with different types of immune responses, depending on what they've been dosed with. People who received 2 doses of the original Wuhan variant, people who haven't received that, but only got their first one of this new reformulated. And I guess that happens with flu, but it's not ideal. And so I could see, for as long as possible, trying to just stay with the same spike. But yeah, I think these are all really interesting questions that CDC, FDA, the vaccine manufacturers have to decide.
So Caitlin, you have some questions in your question thing.
Yeah, absolutely. And I can read them, or if whoever wrote them wants to wants to say them out loud too, but I'll start at the top with just the most liked one. And just click on the thumb if you want to like it. Or Emily, do you want to say your question?
Yeah, I can clarify. I guess I was reading an article that you were quoted in. It was like discussing pan coronaviruses.
Yep.
And I was just wondering what like makes something a pan corona vaccine. If, you can't, like, I guess it just depends what you tested against. So if it's like it worked against like 2+, so now it's pan for the pan you tested on. So that was kind of my question.
Yeah, so this is something that we're really engaged in now, and we're getting funding from Bill and Melinda Gates Foundation, and they're funding a lot of people. NIH just put out a huge call for pan coronavirus vaccine development. And a big issue is that nobody knows how to define pan coronavirus, right? So, and that's what I'm trying to work with Gates to, like, if we could all have this similar panel and similar definitions, that would help. And so I think we could start smaller and think of pan SARS-CoV-2, right? Something that works against all current and future variants of SARS-CoV-2. I think that's very doable. Then you could branch to pan sarbecavirus, so all SARS-like, SARS classic, SARS-2, and other WIV1. That's probably doable. Then you get to pan beta coronavirus, which I think is probably the ultimate goal. If we could do pan beta COVID, that would be really exciting. So that would be MERS, SARS, HKU1, OC43. And then, I guess even more ultimate would be all coronavirus, alpha and beta COVs. But I think if we could limit it to the beta COVs, that's kind of like our moonshot. It's difficult though, and how to go about doing that. I think there's probably 4 ways of making a pan beta coronavirus vaccine. One is just mixtures, a multivalent preparation of spikes. And that could be 10 purified spikes, 10 different mRNAs, each coding from somewhere on the phylogeny of the, I think that's pretty easy. That's probably one of my top ways of doing it. It's just, there's not much intellectual, like just mix them. Something similar, but mixtures of the receptor binding domain, RBD nanoparticles, things like that. A third, which is one we're focusing on because I think it's hard, is S2 only, focusing on like removing the S1 cap and just immunizing with S2. And maybe also cocktails of S2s, but we now have some where it's just prefusion stabilized trimeric S2 only. That's a really cool reagent that we're having fun with now. And I think the 4th way is a T-cell based, like pure T-cell based, and maybe including non-spike antigens, and just trying to get really broad coverage. And the problem is with all of those really is having the reagents on hand, having sufficient viruses to test the breadth, right? So if your vaccine has 10 different spikes, if you do a nude assay against the viruses from which those spikes were derived, that's not really fair. That's not really showing breadth, because that's just like a homologous challenge. So then you need 20 or 30 different live viruses or pseudoviruses to test the protection afforded by 10 different coronaviruses that weren't included in your mixture. And then another complication is that possibly a pan beta coronavirus vaccine will function more at a level of protection versus neutralization. So things like T-cell, S2 only, we have antibodies that can completely protect, but don't neutralize at all. We don't totally understand the mechanism, but that's harder because now you need actual challenge experiments. And live virus, you need mice that are susceptible to each of the different. So it's going to take a while to test those things that aren't based purely on like newt of a pseudovirus.
Thank you. That was a lot of words. I don't think I understood all of them.
Oh, I'm sorry. Yeah, sorry.
That was good.
Thanks.
Anas, do you want to ask your question?
Yes, thank you, Caitlin. So I'm wondering if there are a difference between the immunity which is induced by vaccines in general and RNA vaccines, and the immunity which is induced by natural infection. Are there any experimental evidence showing, for example, that immunity which is induced by vaccines is more robust than the one induced by natural infections, for example?
Yeah, I think those data do exist. And there are multiple studies on that. I think to summarize, the quality of the immune response is similar in that you're generally targeting similar parts of the spike and making similar types of antibodies. And I think Michelle Newsensweig's group isolated B cells from naturally infected versus vaccinated and isolated antibodies and showed that they're similar. I think with the vaccinations, you're getting a more robust and homogeneous response compared to natural infection. With natural infection having orders of magnitude range in your immune response, depending in part on whether it was an asymptomatic infection or hospitalized in severe disease, whereas the immune response from the vaccinations are really reasonably clustered together. It's pretty robust and similar. And so I think that's an important consideration when people are like, well, I was infected, so I don't need the vaccine. They have no idea what their antibody titers, and they could be 3 logs varying from somebody else. But it does seem like maybe regardless of the prior infection, getting like one shot of the mRNA vaccine really puts you high and does a good job of getting you close to like 2 doses of the vaccine. So it seems like anybody who's been infected previously, mild or otherwise, really benefits from getting at least 1 dose of like the mRNA vaccines.
Okay, thank you. That makes sense.
Hi, Lauren.
I have a question, Caitlin, if you don't mind. Sure.
Go ahead.
Yeah, so it seems to me that we've been rather fortunate throughout the last year and a half that pediatric cases have been mild.
Yep.
And my background in molecular biology, I haven't really dug into it a whole lot because I've been away from the bench for a while, but is there a difference in receptor, right? Is there an aging process where ACE2 is expressed more as you get older, or is the innate immune response of children just better, or and do you suspect this is going to be changing as we continue to have more virulent or better expressing variants come down the pipe?
Yeah, those are great questions. And it's an area of active investigation as to why children seem to be relatively well protected from severe disease. There's some thoughts that they've had maybe potentially more recent infections for some of the circulating common cold coronaviruses. But yeah, definitely things, perhaps ACE2 expression levels, TMPRSS2, the protease expression levels. I have young kids, so it's been really reassuring that it hasn't affected them too much so far. Presumably that could change at any point. I mean, this is a large genome. We're really focused on a lot of the changes in spike, but all these variants have non-spike changes in different open reading frames that affect innate immune responses. And so those could be some of the most concerning, where maybe a new variant goes from 1% case fatality rate to 5 or 10, which would be devastating. So I think something that's really under investigation. And I think so far we have been lucky in that most of the changes, the D614G, the original one, and these, it seems to boost titers. I think there's some reports saying like the Delta variant, you're just getting even higher titers in the nose, so it's easier to transmit. But we haven't seen too much of an increase in case fatality rate. Although maybe anecdotally, some people are saying that for the Delta variant, they are seeing younger people, adolescents in the hospital, and people going to the hospital are passing quicker. But I mean, that's just been some anecdotal reports.
Spencer, I know you have to run. Do you want to go with your question?
I don't have to run unless I get called into that meeting. I don't think I need to be.
Never mind.
But I will still ask my question. Jason, do you think you could I don't know how much you've given any thought to this, but I know I've seen reports now that like Moderna is going to start looking into mRNA vaccines for a bunch of other diseases other than coronaviruses.
Oh, they already have, right? They've already had 10, 15 different vaccines in their pipeline, some of which have already been in humans before the coronavirus one.
Okay, so I guess how do you foresee mRNA changing maybe the landscape for influenza specifically? Like, do you think vaccine efficacy for influenza with the mRNA vaccine could be as high as what we're seeing with coronavirus?
Yeah, that's a great question. I mean, yes, some people I think have misconstrued what advantages mRNA has, right? So it does have advantages. One is like manufacturing speed, and so it's fantastic for pandemic response. It's potentially really good for multivalent vaccines because if you were going to include 4 different spikes or 4 different influenza strains, that could be 4 different lots. Or if you're doing proteins, it's 4 different stable cell lines and QC, whereas the mRNA is all the same. So that's really good. And so it's not going to work for everything. Like people are like, oh, now it's going to cure HIV. It's like the problems for HIV are completely not addressed by mRNA, but you could imagine it working really well for flu, and that perhaps they could wait longer before deciding which of the strains they want to include in that year's seasonal vaccine, because there's like shorter time to manufacturing. And then possibly maybe they could go, I think most of the vaccines are trivalent or tetravalent. Maybe they could up it 5, 6, 7 valent, giving them a better chance of not missing and not having a good match.
Thanks.
Mark, did you want to give your question?
Sure, thanks. Jason, just to go back to what you were talking about earlier with the attempts at a pan coronavirus vaccine based on the S2 stabilized. What's sort of your timeline going forward? I know it's hard to predict, but do you have a sense of when that might go into, say, animal trials, or when you might have something that you're ready to publish results on, or what's the progress look like on that?
Yeah, it's going pretty fast. We're really happy with the latest one that Qinglin, the postdoc in my lab, made. It looks great, and we have EM of it, and so we plan to put that into mice probably within a month or so. We'll see how that looks. We'll need challenge experiments, possibly start to write that up if it works. If not, we'll have to iterate and try other things. But yeah, that one looks really exciting. So we're pleased with that S2 only. And we have some other ideas to broaden it, in which case we might need combinations of S2, or we have some weird things like SARS-CoV-2 globular S2 with then parts of other coronavirus S2s like stapled in like a chimera. So we can make those, and we can make nanoparticles. So I think it's going to go pretty quick, but it will be a lot of testing. So I think we'll either fail quickly or succeed quickly.
And what you're studying now is just a subunit vaccine, essentially.
A subunit vaccine for now, but then we could also, we got in touch with people at Houston Methodist, which have like an RNA manufacturing facility, and they've been open to manufacturing like GMP mRNA and lipid nanoparticle. It's funded by CPRIT, so it's like, I think it's heavily subsidized for cancer-related research, but they're pretty keen to try it for like infectious disease. So that'd be really neat. So that could give us our first chance to start trying some of our antigens via mRNA delivery.
Nice. Thank you.
Rachel, I think I saw your hand next.
Yeah, so I wanted to go back to, you mentioned in severity of disease in adolescents, there's a thought that it might be affected by their exposure to common cold coronaviruses. Mm-hmm. So I was just wondering if people have seen any evidence that people who are previously infected with SARS or MERS show cross protection. And part 2, if so, if a pan coronavirus vaccine could have a synergistic effect on efficacy. Yeah,
so I don't think anybody has studied previous exposures to SARS and MERS just because of how rare. There's only 8,000 people in China. that were infected with SARS and for MERS. We've tried doing work with MERS, and it's impossible to get samples like out of Saudi Arabia and other places. So that's been tough. But there are 4 common cold coronaviruses, and there are data from Galit, Alter, and others that showed that a rapid response to S2 upon natural infection seemed to correlate with better survival outcomes, that people that are maybe more recently infected with the common cold virus and had some of these cross-reactive S2 directed antibodies fared better. That's one of the motivations for us to focus on S2. It is the most conserved part. We do have an antibody that can bind both the SARS S2 subunit and the MERS S2 subunit. Those are really quite different, but we at least now have proof of concept that you can get single antibodies binding 2 different lineages of beta coronaviruses. And it actually protects against challenge. It doesn't neutralize, but it protects mice challenged with either MERS or with SARS. So we think that's pretty exciting. Overall, I think a pan coronavirus vaccine, it will never be as good as a virus specific vaccine. Like anytime you go for breadth, you're losing potency. But I think the idea that I have is that if you could make a vaccine that was maybe 60% efficacious against almost all 90% of beta coronaviruses and have that stockpile, during a future outbreak, you'd have a vaccine ready to go that provided some benefit to blunt the first 6 months of the outbreak while the mRNA was ramping up for that specific viral variant. And so that's kind of my goal.